So hello everybody. My name is Kristina Beck, and it's a real pleasure for me today to talk about some of my research on exploring the effects of local population density on social network structure in great tits. And so currently I'm actually based at the Senckenberg Biodiversity and Climate Research Center in Germany. But all the work that I will present today is actually part of my previous postdoc, which was at the University of Oxford, working together with Ben Sheldon and Josh Firth. And so generally in my research, I'm just really interested in animal social encounters. So from really basic social associations where two individuals simply overlap in space and time, up to proper social interactions where two individuals, for instance, groom each other or mate. And a really nice tool to illustrate animal social encounters are, of course, social networks. And I'm sure you here are all aware, but in a social network, these nodes, they represent different individuals, and then the lines in between them show you some form of social connection. So it can again show just a spatio-temporal overlap between two individuals or that two individuals, for example, mated. And so across the animal kingdom, we see quite a lot of variation in social network structure. So not only between different species, but also, for example, if you look within populations. And over the past years, there has really been lots and lots of research showing that these social connections among individuals have consequences for all sorts of different social population processes, such as, for instance, the transmission of diseases or information. And also myself, during my PhD, I started off exploring consequences of social networks, in this case on reproduction. But more recently, I started to become a bit more interested in trying to better understand what the factors and mechanisms are that actually shape social network structures in the first place. And so here we can obviously think about lots of different features, such as the composition of individual phenotypes or things like social inheritance. But one feature that I started to be interested in now is a really, really basic one, which is simply the number of individuals in a given spatial unit that actually sort of build and form the social network. And in this graphic to the left, you can see an increase in population size and density. And so you could imagine that this increase maybe also increases social encounter rates between individuals, and thus potentially leads to also denser social networks. So meaning that we actually have a higher proportion of realized social connections. But at the same time, we can also imagine scenarios where this actually leads to the opposite. So where an increase in population density leads to a decrease in network density. And that's because a lot of individuals are territorial, or they kind of try to avoid each other. And so in such a case, increase in population density does not necessarily have to lead to more social connections. And so, yeah, a lot of different ways in which this could go, this relationship between population density and social network structure. And this is why I kind of wanted to explore this a bit further. And to do so, I investigated great tits. They are a small songbird species that breed socially monogamous during the springtime. So they become territorial. And then during the non-breeding time, so during winter, this territoriality dissolves, and then they forage in fission fusion flocks together with other species. And these flocks, they vary quite a bit in size, and so that's why we kind of see quite some variation in local population and densities. And the population that I studied is located here in the UK, in Wytham Woods, close to Oxford. This middle picture shows you a snapshot of the study site, and then these black dots, they show you the locations of 65 automated feeding stations. And these feeders, they were placed there for 3 winters, about 10 years ago now. And what is special about this population in Wytham Woods is that the majority of birds are equipped with so-called pit tags, which you can see in this picture down here. Yeah. And so this means whenever a bird wearing a pit tag lands on one of these bird feeders, its identity is getting recorded. And so we exactly know when an individual is foraging where and also together with whom. And these feeders were then active each weekend from December to February. And so if we now focus on one of these single locations and on one weekend, what we can nicely see is quite a bit of variation in terms of the local population density. So that is the number of unique gray tits recorded at a feeder at a given weekend. So on average, there was about 15 individuals, but it could also just be 2 birds up to 77 birds visiting. And this is also quite consistent within and across years. And at the same time, we can, of course, now for each of these locations also create social networks from the birds foraging associations. And so in this case, we got more than 2,000 local social networks of more than 1,800 individual gray tits. And so this provided a really nice data set with which we could then sort of explore whether variation in local population density actually affected some of the social network structures that we observed. And so what we found, so we had a look at a couple of so-called global social network metrics. So these are metrics that sort of characterize the overall social network structure. So I'm just showing you 3 different ones here. So one was network edge density. And I put up an example network to the left. So the network on the top would show you a network with a really high density, simply because like every individual is connected to everyone else in this network. And then in the bottom, you have a network with the same number of individuals, but much fewer social connections, so it's less dense. Then the second one is global clustering coefficient. That's a measure which is positively correlated to network density in the case of gray tits, and simply shows you how tightly clustered individuals are within this network. And then the third one would be the average edge weight, which simply shows you how strong all the existing connections are on average in an observed network. And so if we plot these now against the local population size, this is what we find. So generally, for network density and also the clustering, we see somewhat an increase with population size, so meaning that networks became more dense and also more clustered. Whereas for the average edge weight, we see the opposite. So it's decreasing with size, meaning that on average, all the existing social connections become weaker with increasing population size. And so that was really interesting for us to see. But then what we wanted to do as a next step was to kind of compare this observed relationship to a theoretical relationship between population density and social structure. Because obviously in these observed social networks, I mean, they're still sort of shaped by, for example, a lot of different social features. But we kind of wanted to know how population density on its own would sort of affect observed social structure. And so to do this, we performed something that we called a simple model. So a model where we tried to really remove any of the social features. And with social features, in the case of the gray tits, I mean here that we know that these birds really like to forage in groups or in flocks. And also we know that they have social preferences, so preferences with whom they kind of want to forage, for instance, future breeding pairs. And so in the simple model, we really wanted to kind of remove all these features by just picking a given a given number of individuals and then kind of simulating a certain number of individuals visiting a bird feeder. What is the resulting social network structure that we see? And then also as a second model, we ran a so-called social foraging model. So here in this case, we kind of remained the bird's preference to actually forage in flocks, but only removed their social preferences. So for instance, between breeding partners, by using permutations, we kind of disrupted all the social relationships between birds. So for both these models, we then calculated again these global network metrics that I was mentioning before. And then we compared them to the observed data. And so I'm only showing just one example for one of these metrics, the clustering coefficient, but it looked really similar to network density, for example. And what we can see across the three different data sets is that the relationship between clustering and population size is somewhat similar. So we see somehow an increase, at least sort of at the initial population sizes. But then if we compare sort of the clustering coefficients that are estimated from the simple model with the one from the observed data, we see that they are much, much lower. So it really underestimates the clustering that we actually observe, which to us suggests that really these social features are kind of important in generating the clustering that we observe. In contrast, also, when we now look at the social foraging model, it fits the observed data much, much better. But we see that it actually overestimates clustering, which again, in this case now suggests that, well, the bird's tendency to forage in flocks is really important in sort of generating this overall social network structure. But then also these distinct preferences between birds are really important in actually shaping the clustering that we observed. And so to just quickly summarize this study, so we could show that population density affects global social network metrics, but also that much of the observed social network structure is actually still shaped by different social processes, such as the bird's tendency to forage in flocks, and also different social preferences between the birds, such as, among social partners or breeding partners. And so I was really interested in that because given that across the study side, we have this really consistent variation in local population densities, this potentially means we have also consistent variation in, the social connections that we sort of see locally. And this can now obviously also have consequences for different social processes. And so one social process which is really studied a lot in the literature in relation to population density is actually the transmission of diseases. And so here very often the logic is that, well, if you have higher population densities, you get more social encounters between individuals, and therefore you also get more opportunities for diseases to spread. And since I was working on social learning at that time, I was wondering if maybe the same logic applies to the transmission of information. And to kind of test this, we thought it would be really neat if we could actually experimentally manipulate local population density. And to do so, we used, again, this RFID system that I mentioned at the beginning. So a majority of birds, they are equipped with these pit tags here. But in this study, we used a different type of bird feeder, a so-called selective RFID feeder, which means that it can selectively grant access to certain individuals. So if a graded with a pit tag lens on the bird feeder, we can either sort of allow it access to the food or we can deny access. And by using the system, we then were hoping to sort of be able to manipulate local population densities. So in a bit more detail, what we did, so this study was conducted from January to March in 2021. And so we set up 6 experimental sites shown in black, and then 2 control sites shown in white. And within each of these experimental sites, we placed 2 of these selective bird feeders about 100 meters apart. And then at one of these bird feeders, we wanted to create this low density condition, and at the other one, a high density condition. And so in a bit more detail, so at each of these experimental sites, when we placed these bird feeders, we first had a pre-experimental period of about 2 weeks, where there was no restriction. So every bird could just freely forage at any of those 2 bird feeders. And then the experimental part followed for about 6 weeks, where then at the low density bird feeder, we only provided access to 20% of all the previously recorded birds. And then at the high density bird feeder, we excluded those 20%, but allowed everyone else access. And we were hoping that this would then sort of create a low and a high density condition. And then finally, we of course wanted to see now whether this had any effects on information transmission. And to do so, we ran a so-called patch discovery experiment, which means that we placed a novel feeder close to both the low density and the high density feeder. And then we recorded the individual's discovery of this novel food source. And that's an experiment that has been already done a couple of times in the past, and where we know that information about these novel food sources transmits socially. And so this is what we found. So first, we were, of course, interested to see whether the experiment even led to the desired changes in population density. And for this, we first focused on the local level, meaning that we always compared the low density feeder with the high density feeder. And across the remaining talk, everything you see in gray is referring to the low density treatment, and everything in orange to the high density treatment. And some of the things we had a look at was the proportion of recordings and also the number of individuals visiting each of those bird feeders. And so what we found was that, well, there was no difference in the 2 prior to the experiment, but then during the experiment, we had a higher proportion of recordings and also a higher number of individuals visiting the high density feeder compared to the low density bird feeder. Similarly, if we focus on the individual level, so here we always compared birds in the low density treatment to birds that we assigned to the high density treatment. And we see that there was no difference in sort of the average flock size sort of prior to the experiment, but then during the experiment, birds in the high density treatment on average had larger flock sizes than the birds in the low density treatment. And then second, we were interested to see whether these changes in density also again led to changes in local social structure. And here again, first focusing on the local level, we created social networks for each of these bird feeders separately, so daily social networks. And then we extracted again a couple of global network metrics. So similarly to this first study that I was talking about. So for example, we had a look at network density and the clustering coefficient again. And what we find again is no difference in these network metrics prior to the experiment, but then during the experiment, networks at the low density bird feeder were on average less dense and less clustered, which was quite nice to see because it was sort of similar effects that we actually found in this first study. Again, we were also interested in individual level changes in sociality. And so here, instead of calculating social networks for each bird feeder separately, we calculated one social network considering the foraging associations at both the bird feeders. And then we extracted a couple of individual network metrics that sort of describe an individual's position within the network. So for example, we had a look at the weighted degree and the weighted eigenvector centrality. So they both kind of show how social and centrally positioned an individual is within a network. And again, what we find is no difference prior to the experiment, but during the experiment, birds in the low density treatment, they were on average less social and less centrally positioned. And now finally, we of course then wanted to see whether this had any effects now on the discovery of these novel food patches that we had set out. And so in this case, I first wanted to see whether individuals in the high density treatment, whether they are maybe more likely to discover these novel food patches, and also whether they might be faster in discovering these. And yeah, just to make it short, we didn't find evidence for this to be the case. So if we focus here now on the patch discovery experiments during the experiment, we see that there is no difference in the likelihood and also sort of in the order in which birds sort of discover these food sources between high density and low density birds. And yeah, so now to sum up this study as well, I think it nicely shows how automated feeders can be used to experimentally manipulate local population density and local social structure. It again also shows how changes in population density can affect social structure. And we found no evidence for effects on information transmission. And this could be due to all sorts of different reasons. But one that we kind of assume is because these changes that we generated in the social environment, specifically for individual sociality, they were actually relatively small. So if we looked at things like the average flock size, I think birds in a high density treatment, they had on average just a couple of social connections more. And so maybe that was just not meaningful enough to sort of create a meaningful difference in terms of the information being transmitted about these novel food patches. But yeah, it could also be all sorts of other reasons as well. And yeah, now at the end of my talk, just to sort of conclude from these two studies, I hope that I could demonstrate that variation in local population density can affect variation in local social structure. And even though we didn't find any effects in our experiment on information transmission, I think it nicely demonstrates that it could potentially have consequences for all sorts of different social population processes. And I think for me, this of course now really begs the question of what is sort of causing variation in local population densities in the first place. And again, there's several different features we could think about, such as emigration, immigration, or the habitat configuration, such as the vegetation structure. That's actually also something that we had a look at in this first study that I mentioned, and just didn't have time to talk about here today. And also one feature that I had to think about was human alterations, because there's really lots of human activities that sort of modify the environment individuals live in, and which impacts local population densities. So things like hunting or pollution can drastically reduce population densities. but then also many other activities which can really increase densities. And one feature here that I had to think about a lot was anthropogenic food sources. And I just briefly wanted to mention this at the end of this talk, because this is what my current project is about. So to kind of explore the effects of anthropogenic food sources on social structure. And I'm sure we can all think of all sorts of different examples of anthropogenic food sources, so from landfills to fishery discards, or even like the artificial, bird feeders that I was mentioning as well in my studies, or sort of directly providing food to wildlife. I think they can really influence animal social encounters. And this is specifically because I think in comparison to natural food sources, they are often highly abundant and predictable, and also often in close proximity to humans. And I think those two features probably can have really interesting effects on animal social encounters. And to explore this further, I set up a project on alpine choughs in Germany. So they are a corvid species which lives in alpine environment, and they are highly social. And why I started to be really interested in them is because they really use a whole lot of different anthropogenic food sources. So from scrounging on food leftovers at mountain hearts to landfills, up to really directly approaching, humans and scrounging some food there. And so, last year we equipped a couple of birds with GPS tags. And so we're hoping to kind of explore a bit further how these different anthropogenic food sources actually influence social encounters. So hopefully in the future I can tell you a bit more about that. And with this, I just really like to thank a whole lot of different people that were sort of involved in these two studies that I presented today. And all of you, of course, for your attention.
Thank you for this very nice talk, Kristina.
You're welcome.
So I open the floor for questions. So just unmute yourself and jump in.
Okay, I'll jump in.
Great.
Good. Thanks a lot for the awesome talk. I was wondering, firstly, the manipulation that you did for the experiment was super cool to try to vary the population densities. And you said the reason you didn't find a result could be because of a lot of things. I was wondering if you just talk about what those a lot of things might be.
Yeah, sure. Where to start? Maybe first to start with, things considering the setup. So maybe you remember that I was showing that we put those novel bird feeders like about 40 meters apart. And so the reason why we chose 40 meters is because 40 meters is kind of far away enough so that the birds don't see them while they forage on their familiar bird feeders. But it should be close enough that they sort of can discover them fairly easy. But what I think could also, or what could have been one of the reasons, is that it might have been too close, so too easy for the birds to find, in which case, no kind of social information transfer was sort of necessary, because birds, just really easily sort of discovered them. And so the manipulations in terms of their social network were just not really, meaningful enough. And then, of course, you could also think that maybe information, just didn't transmit socially in those cases. Could also be that case. But as I was saying, I mean, I really think that one of the main reasons could really be the social environment, and that, because we only had this density manipulation running for about 6 weeks. And so I could imagine if you run this through like the whole winter and really sort of establish these local differences in the densities, that maybe the picture would also be different. Because what we could also see, for example, or I mean, I didn't look into this very specifically, but when we split up the birds into these different conditions, we didn't consider things like already established social pairs, for example. And we know that the future breeding pairs, they really sort of prefer their social relationships over if they get their food access or not. And so I think that's also one of the reasons why maybe these differences that we discovered in changes in individual sociality, which is not as big as we were kind of hoping to. And yeah.
Sure, totally. Thanks a lot. And I had one more follow-up question.
Yes.
So without the manipulation, I was wondering if you looked at the differences in densities between different points in the patch in across Whiteham, and if there are any patterns that you found where birds naturally hang out.
Yeah. So I briefly mentioned at the end of my talk that in this first study that we had a look at vegetation structure. And so this was actually not data specifically collected for our study. So these were some botanists a couple of years ago, I think it was 2012 or so. They collected data on shrub cover density. And shrub cover is something that the grey tits really like because it sort of gives them protection while foraging against predators. And so we kind of assumed that, well, in those areas where you have really dense shrub cover, you maybe also get a higher population density. And then also one of the things that just by plotting, to me, it kind of looked as if population densities are larger, sort of at the outline of the study site. And what is maybe important to mention here is that outside of the study area, so outside of Whiteham Woods, it's all agricultural area, so that it's not like forest continuing, it's all agriculture. And so this is actually what we found. So population density increases with sort of is like higher at sort of the border or outline of the study site. Which, is really a bit puzzling. I mean, we found sort of no significant effect of the vegetation structure, but it was sort of a trend towards the right direction. So I'm thinking if we would have like better data specifically collected for our purpose, that we could find that, vegetation plays a role in sort of predicting, some of the local population densities that we see. It's always tricky if you just take some data set which was sort of collected for a different purpose, and also it was a data set that was only collected in one of the years, and so the data that we analyzed was 3 years, and especially things like shrub cover density can potentially change quite a bit as well. And so it wasn't really ideal data, but it seems that the sort of the boundaries. So the forest edge, basically. So densities were closer at the forest, were higher at the forest edge. So that's something we found.
Thanks. Yeah, it kind of matches my intuition, although I never looked closely at it. But also the tits in the German patch also seem to, like, you don't find very many of them in the center of the forest, but they're all just around the edge as well. So yeah. Okay, cool. Thanks a lot.
Yeah, sure. You're welcome.
Hi, Kristina. Thank you for the presentation. Hello, can you hear me?
Yes, I can hear you.
Thank you so much for the presentation. Super cool, especially the automated feeder. It's just super cool. I actually have since I don't do animal research, so I'm like more curious about your modeling part. Like in your first study, you mentioned you did a model first. It's a simple version, and then it's one with social foraging. Could you please tell us a bit more how specifically you did the model? Is that like a simulation?
Yes, I'm not sure if I could hear everything, but you just wanted more details about these models, right? Like the
Mhm,
simple
yeah.
model and the social foraging model. Yeah. So for the simple model, what we did is basically just we first decided on a given number of individuals, which sort of reflected also the number of individuals that we had sort of observed from our real data. So I think at the beginning I mentioned that it kind of varied from either just 2 individuals visiting one bird feeder at a given weekend up to like 77 birds. And so, for instance, in one simulation run, we would take, I don't know, 20 individuals. And then what we did is that let's start differently. So what we then did is for 20 individuals, we just randomly picked 20 out of our observed data and then selected also these foraging times, right? So we had 20 random individuals with distinct foraging times and also distinct number of times that they foraged. And then we put those together at sort of one, or mimicked as if all these birds were visiting at these distinct times at one location. And then we just created a social network again. And that's because we kind of wanted to remain the timing during which birds could potentially visit sort of constant, and just kind of try to mimic, or kind of change the number of individuals, but sort of remain this timing during which they visit constant as well. That's why because we could have also said like take a number of individuals and let them visit during random times. But yeah, that's why we kind of decided to go with these natural times that we also observed. And then in the case of this social foraging model, so something that we observe in the social networks is that individuals kind of have preferences for with whom they actually like to forage. So if we take one bird feeder, what we see is that we get a couple of bird flocks sort of arriving at this bird feeder at a given day, for example. And then so in this case, we used randomizations. So here we just so if you imagine you have like flock A, flock B, flock C, which always gives you a couple of individuals that visited during a specific incident. And then we were just like reshuffling individuals across these different flocks. And by doing so, we kind of remained the flocking structure the same, but we broke the specific social relationships between the birds with whom they kind of wanted to forage with, if that makes sense.
Yeah. Yes, I think it's super smart. Yeah,
Oh,
is that
thanks.
a typical like a typical method in your field, or you
Yeah.
this method? Oh, is it like a
Yeah,
typical
yeah.
method?
Yeah, it is. So in social network like analysis, it's quite often used that you have these permutations, and because it's really often like an issue, like trying to disentangle, is it really, some kind of social behavior or like a social preference between different individuals, or maybe they just have the same spatial preferences, right? So you could imagine that birds just really prefer the same spatial location. And that's why in your social network, they also appear as if they would be socially preferring each other, but actually it's just a spatial preference. And this is why in social network analysis, we use a lot of these permutations to kind of try and get to the bottom of this, like, is it really some kind of social preference, or maybe just really a spatial overlap in a sense. Yeah.
Gotcha, thank you.
You're welcome.
So if no one else has a question, I jump in.
Yes.
I was wondering if you also looked into not only the density, but also in the social structure, like which age types were together in a flock, if this changed your simulations, or maybe also different personality types.
Yeah, so to make it short, I did not, but it would be really interesting, of course. So one of the, I mean, initially we kind of had ideas of maybe also considering, you know, different compositions of individual phenotypes, and it was just getting way too complex. So we kind of thought, well, let's just jump back and just focus on, you know, the number of individuals for now. But of course, I think that that would be a really crucial next step, like not only to look at the density or the number of individuals that you have at a given location, but really also the composition of individual phenotypes. Because we know, for instance, that younger individuals, they tend to be sort of more social compared to sort of other individuals or have more social connections. And so that will obviously also really contribute towards, you know, these observed social networks, and be also meaningful then for all these other social processes, like how information transmits and so on. So yeah, a lot more that could be done with that data set. Yeah.
But I can imagine that it's getting way too complex very easily. So
yeah, totally. It was, yeah, I really have to always laugh about these studies, because you often start off with discussions and have all these ideas about what else you could sort of, yeah, put together. And then, I mean, at least in my case, like I often sort of then start splitting projects, right? I start off with one big one, and actually it ends up in two different papers. But at the initial stage, you think, oh, you can put all those different things together in one study, and then you soon realize, oh, well, this is getting too complicated. We have to reduce somehow. But yeah.
thank you.
Hey, sorry, could I build on that question as well? Again, I think what you guys were chatting about there was super interesting and fantastic presentation. I came in a bit late, but I was just wondering if, based on what you were just discussing there, and now your future work, looking at some of these like people's personal bird feeders in their gardens that are perhaps a bit more of a stable resource, whether any of that, then whether you could use that to dissect some of that more like long-term relationships that have lasted over time. If they're coming back to a regular place, are they coming back there like every summer, or things more like long-term stable social networks in that regard?
Yeah, totally. So in my current project, so I started now establishing this new project on this different species, the alpine chaff. But I'm already thinking about new projects potentially on the tits, simply because, you know, like experimental work is just, you know, much more easier with these birds. Yeah, it's really funny that you were mentioning like people's gardens and stuff, because I'm kind of thinking about new projects where I can exactly use this, or also because gray tits, they really nicely occur at this gradient of human activity with, you know, yeah, people's gardens, or really in cities, and then from villages to more remote places. So I think, yeah, it opens up like all sorts of new and interesting ideas, and to sort of properly look into that.
Yeah. No, thank you.
There are no more questions. I think we grilled you enough. Thank you very much for this nice talk. And
you're welcome. Thanks for inviting me. It was a pleasure.